Hello everyone, my name is Yeon Seo. I'm a master's student at Sungseo University in South Korea. Thanks for joining me today. This is overview of today's presentation. I walk through background, methods, objectives, results, conclusions, and future study. The field of immunotherapy can be categorized into four main approaches: immune checkpoint inhibitors, adoptive cellular therapy, cytokine therapy, and cancer vaccine. Unlike traditional approaches such as surgery, radiotherapy, and chemotherapy, immunotherapy offers higher specificity and long-term anti-tumor effect. And most importantly, immunotherapy stimulates the patient's own immune system. Today, I'm just focused on adoptive cellular therapy. Adoptive cellular therapy, or ACTF, evolved since the 1980s. So in the first early, we saw the development for generation ACT, especially tumor-infiltrating lymphocyte. In the 1990s, 2010, the second generation ACT, especially CAR-T therapy, gained a lot of interest. And now we're in era of third and next generation ACT, especially TCR-T therapy. T-cell therapy market is expected to add growth rate factor of 37.6%. It is driven by increasing approved products and expanding manufacturing capacity. For T therapy, first generation ACT, we isolate tumor-infiltrating lymphocytes from tumor mass. And these T cells are expanded ex vivo, and after preconditioning, these T cells are injected back into the patient. And for TCR-T and CAR-T therapies, third and second generation ACT, we isolate T cells from peripheral blood, and these T cells are genetically modified to recognize tumor antigen or patient-specific neoantigens. Follow T cell expansion and formulation, these T cells are injected back into the patient. CAR-T therapy works by mimicking the natural process of T cell activation that acquire instinct three signals. In signal one, binding between TCR and peptides MHC molecule. In signal 2, costimulatory domains signals such as CD28 and CD88. In signal 3, cytokine signals. So we can engineer single-chain variable fragment to recognize tumor antigen, and it is connected to a costimulatory domain and CD3 zeta domain. So engineered T cells are expanded ex vivo and injected back into the patient. So for now, CAR-T therapy, 8 CAR-T therapy have received FDA approvals. But CAR-T has its limitation. First, it can be only detect surface antigens that make for 25% of human proteins. Second, its clinical success have limited to certain blood cancers that make for 5% of all cancers. Finally, CAR-T therapy has lower antigen sensitivity than TCR-based approaches. So to overcome these limitations, TCR-T has evolved. TCR recognize peptides and MHC molecules and that allow targeting diverse antigens of neoantigens and viral proteins. Second, unlike CAR-T therapy, which is restricted to certain blood cancer, TCR-T can be applied to solid tumor treatments. And finally, TCR allows as few as 1 to 50 pMHC molecules compared to the thousands of antigens needed for CAR-T therapy. But TCR-T has its limitation also. First, because TCR consists of alpha and beta chains, so engineering TCR pairs is technically demanding. And second, it's difficult to handle the tumor microenvironments that suppress T cell activation. Finally, most importantly, is extremely difficult to identify TCRs that are both antigen-specific and self-tolerance, that critical requirements for safety and efficiency. So we just focus on third challenging to generate epitope-specific TCR sequences. Our goal was to generate epitope-specific TCR sequences, so we had to decide which generative model to use. There are a lot of generative models, but they share a common principle. They run distribution of data and they generate new but realistic samples that follow the training data. So generative models are already being used in a wide range of applications, such as denoising cryo-electron microscope data, single-cell gene expression analysis, and protein design and generation. And now we just decided to use diffusion model because that is useful for image data. So let me explain how diffusion model works certainly. We think of diffusion, we typically imagine molecules spreading over time like this. And let's say we have 3 molecules diffusing over time, and we just focus on this purple one. At each time step, its position changes gradually here, and here, and here. And then let's say we want to reverse this process. If we can estimate how much it changes at each time step, we can reverse this process. So in diffusion model, they treat these changes as noise. So if we can predict this noise, we can reverse this process and recover the clean data. So we can see 2 images becoming noisier over time. And in diffusion model, they run the distribution of data by adding noise and denoising it step by step. So finally generate new but realistic samples from this noise data. This is objectives overview. We aim to use diffusion model to generate epitope-specific TCR sequences. And then we fine-tune this model to expand its capability to predict TCR epitope binding. This is an overview of the TCR-epiDiff architecture. First, we collected TCR and epitope sequences from 3 data sets. And its amino acid was converted into ATGC level and what are encoded like this. And as explained earlier, we use 4 diffusion process by add noise to data, and this noise data pass through this unit-like model. So our model downsamples the input from 4 to 512 dimensions and upsamples to 4 dimensions. And we also use skip connection layers because of preserving essential information like this. And noise level changes at each time step. So we also incorporate this time embedding vector into every layer like this. And most importantly, we wanted to generate epitope-specific TCR sequences. So we used to proteinify Excel to create epitope embedding vector and then injected them into every layer like this. So in the other words, for forward process, noise is added to data. And for reverse process, our model predict the added noise. So for TCR-epiDiffs that generate epitope-specific TCR sequences, so we use 3 data sets. And however, for TCR-epiBP that predict TCR epitope binding, we had to have both positive and negative data sets. So a major challenging is how to generate negative samples, because we use 3 data sets that only contains positive samples like this. So a common approach is to generate negative samples by pairing existing epitopes with healthy PBMC TCR sequences randomly. But this method cannot guarantee completely negative samples because nobody knows these pairs are perfectly negative samples. And we apply these methods random negative data. So to overcome its limitation, we use IDB datasets that contains validated non-reaction antigen and PBMC assay. So we collect these negative epitopes from IDB and pairing with randomly healthy PBMC TCR sequences. And then we apply these methods IDB negative data. And finally, for external validation, we use COVID-19 and neo-TCR datasets. So to summarize, first generation model part, we developed TCR-epiDiff that generate epitope-specific TCR sequences. And then we fine-tuned encoder and added linear layer like this and fine-tuned with random negative samples and ones with IDB negative samples. And then we call this method TCR-epiBP, and we call this method TCR-epi*BP. And most importantly, for TCR-epi*BP, we also incorporate the HLA type information like this. So I will explain about that shortly. When TCR recognize peptides, TCR not bind peptide only to peptide MHC molecules also. So MHC information affect affinity between TCR and peptide. So for TCR-epi*BP, we encoded HLA type information. So we made this one unencoded, and this vector were concatenated every layers like this. So to summarize, for TCR-epi*BP, we fine-tuned TCR-epiDiff encoder with IDB negative samples and also incorporated HLA type information. And now this result, I will explain our result in 5 parts. For TCR-epiDiff, for process, we set noise level beta start to 0.001 and beta n to 0.1. So our sequences becoming noisier over time like this. And for reverse process, after 34 epochs, our models converged and early stopping was applied, lows around 0.001. So we wanted to verify TCR generation quality. TCR-CD3 beta sequence are known to have conserved ends starting with CASS and ends with F or FF. So our generation sequence like this reflected conserved patterns like this. So indicating that our model successfully run the TCR sequences feature. And also, we found higher correlation at sequence N and higher variation in middle of sequences. Also, cosine similarity supported generation TCR sequences were close to original TCR than to control. And next, we wanted to test two external validation sets, COVID-19 and neo-TCR data sets. We apply forward process and this noise data pass through trained models and then followed by 3 denoising steps. On both COVID-19 and neo-TCR data sets, we also found these conserved patterns like this and higher correlation at sequence ends like this and higher variation in middle of sequences. And next, we wanted to visualize 512 dimensions latent TCR using UMAP. For 10 epitopes with 45 through 16 TCRs, we performed KNN analysis and achieved 0.78, indicating that latent TCR target same epitopes clustered together like this because we use proteintify XL that create epitope-specific vector and injected them every layer. So epitope-specific information were well encoded into latent TCR. And next, we wanted to perform TCR-epitope binding prediction. First, we fine-tune TCR-epiDiff encoder, once with negative, random negative samples and once with IDB negative samples. Most importantly, for TCR-epi-P, we also incorporated HLA type information. So for TCR-epiBP, training accuracy was 0.786 and test accuracy 0.743. And for TCR-epi-BP, training accuracy 0.855, test accuracy was 0.855. Indicating that inclusion of HLA type information and fine-tunes with IDB negative samples clearly improve performance. And next, we perform benchmarking against existing model. Our models outperforms all baselines compared to existing models like this in TCR and epitope bind tests. This is conclusion. We developed TCR-epiDiff that generate epitope-specific TCR sequences. And generation sequences reflected the conserved patterns like TCR-CD3 beta sequence. And we also incorporated epitope-specific information. So latent TCR target same epitopes clustered together. So epitope-specific information were encoded well into latent TCR. And next, we fine-tune this TCR-epiDiff and to expand its capability to predict TCR epitope binding. Our model, TCR-epi-P, outperforms all baseline compared to existing models because we incorporated HLA type information and we fine-tune IDB negative samples. So we expect our models can help adaptive cellular therapy field. This is future studies, so we concern about how to integration protein 3D structure information. This is our labs, and especially thank you, Professor Jacqueline. Thank you. Thanks for listening.
Thank you. Any questions? There's one. Yeah. Microphone 2, please.
Okay. So very interesting lecture. One question: when you're doing real kind of next generation sequencing of the TCRs from the TIL or whatever, you will see huge networks with only one amino acid difference. How can you explain in this case the extremely different affinity to completely different targets with only one amino acid difference in the middle of the TCR, CDR3 I'm speaking about? Because this model will not explain that, right?
Okay, it's a really good question. I agree with your question because TCR is variable among individuals, so it's really difficult to identify TCRs among individuals. So it's really hard to select. So, yeah.
And another question: we used the data set that extremely heavily skewed towards the virus CDRs, right? So COVID, all this influenza and stuff like this. So your model, it's actually use initial data kind of that are very heavily trained, let's say, on these TCRs that recognize virus particles. So I think you need to increase your diversity and try to predict the neoantigens and do some real life experiment to see that you can recognize whatever receptors or whatever proteins. So something like this to show that it's can be used in real life, not only as theoretical modeling.
Okay.
We have time, only one more question. Yeah.
Thank you. I think I got lucky. I have a question for your first epidiff model. In the encoder and decoder, there's a latent space. How do you ensure the latent space actually capture the features from your original TCR sequences? Or maybe like, what is the exact loss function to ensure doing this?
Okay, I have thought deeply about your question, so I have to solve in your question. Yeah.
Oh,
Maybe you can talk offline. then.
yeah, thank you.
Yeah, because we need to stick to time. Thank you.
Thank you.
Yeah. The next speaker is